Good morning, everyone. Oh, Good morning, everyone. My name is Adam Lanani. I'm one of the summer scholars in the Mayan Lab, and I'm a rising sophomore at Brown University. Today, I'm going to be telling you about Genes4Networks, a web-based tool to construct protein-protein interaction subnetworks given sets of input genes or proteins. First, I'm going to give you a quick outline of my presentation. We're going to dive into relevant background information about protein-protein interaction databases and a tool that was previously developed by the Mayan Lab called Genes4Networks. Next, I'll dive into discussing protein-protein interaction databases and how I implemented Genes4Networks. And finally, I'll introduce a case study that demonstrates how Genes4Networks can be used for target discovery. It has been estimated that there are between 20 to 25,000 protein-encoding genes in the human genome. And these proteins give rise to between 130 to 650,000 protein-protein interactions, or PPIs for short. PPIs are critical to all biological processes, and new PPI data is published every year via various assays and computational methods. In the chart on the right, we can see the growth in the number of PPIs in the Biogrid database over time. PPI data is a very useful resource for biologists. It provides a critical source of evidence about how the thousands of different types of molecules inside cells come together to produce emerging cellular phenotypes. Genes4Networks was a web-based tool first published in 2007 that originally integrated 10 PPI databases. Genes4Networks takes an input list of genes or proteins and outputs a subnetwork that connects these proteins and also a table with ranked significant intermediates. Genes4Networks, the project we're talking about today, was created as an update to Genes4Networks and incorporates many more PPIs from the most recent publicly available PPI dataset. Before discussing Genes4Networks a little more, I would also like to mention that Genes4Networks was used to discover a gene, namely SHOC2, that when mutated can cause a devastating Noonan-like syndrome. Patients with this disorder suffer from short stature, learning difficulties, and heart abnormalities. The gene SHOC2 was discovered because it only has 4 known protein-protein interactions in 2007. 2 of these are interactions associated with the gene related to Noonan syndrome. SHOC2 was sequenced in 110 patients that had Noonan syndrome or Noonan-like syndrome, but did not have mutations in the previously 4 known Noonan genes. This was all, of course, done before exome sequencing was available. Now, let's get back to Genes4Networks. Genes4Networks contains 4 primary data sources: Biogrid, String, Integrated Interactions Database, or IID, and Bioplex 3.0. Biogrid contains PPIs extracted from over 70,000 studies with both high throughput and low throughput focused studies. Genes4Network includes about 900,000 non-redundant PPIs between about 20,000 proteins from Biogrid. The plot on the right shows a connectivity distribution for the proteins we gathered from Biogrid. It follows the common power law distribution, meaning the graph contains a few connected genes or hubs, while the rest of the nodes have fewer connections. String has just under 12 million PPIs in its database. Due to this large number, we decided to only take from String the highest confidence interactions with a threshold of confidence score greater than 0.98. We're left with 7,000 proteins and about 29,000 non-redundant PPIs. Again, a power law distribution for the plot can be seen. IID is a very large database of about 18,000 human proteins and 1.2 million interactions found through experimental studies, orthologs, or machine learning predictions. The connectivity distribution of this database also follows the expected power law distribution. And finally, Bioplex 3.0 has about 14,000 human proteins and 118,000 PPIs, all discovered with affinity purification mass spectrometry. The connectivity distribution again follows this power law distribution. One of the challenges we had in fusing the databases was the different protein identifiers used across all the databases. To solve this, we converted all the protein names and IDs to Entree gene symbols using the NCBI gene database. We then combined all the PPIs from all the databases into one file, filtered out the duplicates, removed self-interactions, and also removed genetic interactions. In the end, we were left with 20,000 human proteins and about 1.47 million interactions. On the right, we have an upset plot detailing the intersection between these databases. As you can see, Biogrid and IID share many of their interactions. The harmonized data was then serialized and ingested into Neo4j, which is a graph database. The ingestion was done with the help of a script developed by Errol Evangelista, a bioinformatician in the Mayan Lab. Once ingested, Neo4j is connected to a user interface, which is developed in React and Next.js. This UI, which can sit on top of any Neo4j database, was also developed in the Mayan Lab, with Errol being its primary developer. Through the UI, we can input a gene set, and then after pressing submit, the app outputs a subnetwork that connects the proteins from the input list with protein-protein interactions from all the databases. Now that we've gone over some relevant background information on how we developed Genes4Networks, we're now going to demonstrate in action with a case study that demonstrates how Genes4Networks can be used for target discovery. Our collaborators, Dr. Mark Keller and Dr. Alan Lanani from the University of Wisconsin, are world-renowned experts in the field of type 2 diabetes. In this project, they aim to study the relationship between gene expression and insulin secretion. They analyzed data they collected from diversity outbred mice, a collection of over 500 diverse mice that are used as a model for mimicking genetic diversity in humans. The mice were fed a Western diet, which is a high-fat, high-sucrose diet for 4 months. Then, RNA-seq and insulin secretion data was collected from the pancreatic islets extracted from these mice to measure gene expression and insulin secretion level. Here's an example of what that correlation looks like between insulin secretion and gene expression for the gene E4BP4, a gene that is known to be involved in insulin secretion. You can see the gene expression levels across the diverse mice has a high correlation with insulin secretion. Measuring the correlations between insulin secretion and expression levels across all the mice for all the genes was useful for identifying many genes that are positively and negatively correlated with insulin secretion. I took the 30 most positively correlated genes and entered them into Genes4Networks to identify additional genes that directly interact with top positively correlated genes. These additional genes are also likely to be related to insulin secretion. Once these 30 genes were input into the form, we updated the parameters of Genes4Networks by filtering out high throughput and machine learning predicted interactions to increase the quality of our results. We then submitted the set for analysis by pressing the Generate Subnetwork button, and here are the results. On the left, we can see a table of intermediary nodes with the highest z-scores computed by the binomial proportions test. The transcription factors, retinoid X receptor alpha and PPAR gamma, are ranked second and third. This is encouraging because these transcription factors are key known targets for drugs already used to treat type 2 diabetes. On the right, we can see the subnetwork of protein-protein interactions with retinoid X receptor alpha and PPAR gamma highlighted. As mentioned, the transcription factors retinoid X receptor alpha and PPAR gamma form a heterodimer to regulate the expressions of genes that are known to be related to insulin sensitivity and are targeted by diabetes drugs. The results suggest that the many genes identified by Mark and Alan are regulated by these transcription factors. It also suggests that Genes4Networks may be useful for discovering novel drug targets in this and other contexts. To conclude, significant intermediaries discovered by Genes4Networks can be a hypothesis for further exploration. I would also like to mention that Genes4Networks can be accessed via API, and in this way can be added to various other workflows, as you'll see shortly in Hannah's presentation. In the future, we plan to improve Genes4Networks by adding other forms of protein identifiers, adding support for variants, adding gene-gene co-expression data, integrating with the CFDE knowledge graph, incorporating more data from string, and finally, adding new PPI data as it's released to keep Genes4Networks up to date. Now, I would like to make a few acknowledgements. First, I'd like to thank Dr. Mayan, our PI, for the opportunity to learn as a summer trainee and for support throughout the project. Secondly, I'd like to thank Ms. Jenkins for doing an amazing job running this program. And finally, I'd like to thank Errol for his mentorship and his guidance throughout the project. He really helped make this project possible, and I'm super appreciative. Thank you so much for listening. Please leave any questions in the QA section below. And next, I'm going to be passing off to Hannah Lee. We'll talk about how she optimized a pipeline that identifies kinases and transcription factors given sets of differentially expressed genes. Thank you.